The title of this paper is Menstrual Cycle Variations in Gray Matter Volume, White Matter Volume, and Functional Connectivity. There's a critical impact on parietal lobe. I wrote this paper back in 2020, or published this paper back in 2020. I wrote it 2020, 2018, 2019. Takes some time to get through. Review. It was work that I did during my PhD dissertation. It was work that was collected probably in 2007, 2008, I believe. And that'll become somewhat important. Um, so the summary of what we're going to go through is we're going to review the paper. So Meeker et al.'s menstrual cycle paper. This is the 2020 version paper. There's another paper that's from 2021 that I'll also review at some point. We're going to have the background and introduction to the paper, methods, results, and figures. Most of the results are as figures. A discussion, summary, and conclusion. And then that's going to be the end of what you need to do for a journal club presentation. At this point, we'll transition into more recent major results from more recently published manuscripts. Some of these I'll have the figures on, some of them I won't, on the association of menstrual cycle with brain structure and function. An overall summary of the results and possible plans of the lab, my lab, to address gaps in in current gaps in the literature in the future. So the background. So hormones fluctuate across the menstrual cycle. During the menstrual phase. In this study, we divided the menstrual cycle into 4 phases. During the menstrual phase, during menses, the low estradiol and low progesterone. During mid-follicular phase, there's low estradiol and low progesterone, but progesterone is increasing. Or estradiol is increasing, I should say. During the ovulation, there's high estradiol and low progesterone. During luteal phase, there is high estradiol and high progesterone. So there have been numerous studies before this, and they're cited in the paper, most of them. Studies of gray matter in the female brain across the natural menstrual cycle, and these have sort of revealed phase effects in the hippocampus, fusiform gyrus, amygdala, frontal and parietal cortices, kind of widespread. There's a lot of white matter changes that are small but consistent. Studies have also reported estradiol enhances performance on cognitive tasks in rats, mice, non-human primates, and pre- and postmenopausal humans. So why did we do this study? Basically, this study was done when I came into the lab and we had the data. The data was sitting there, and I was like, well, this is an interesting question. I'd like to look into it, and I was allowed to look into it. During looking into the menstrual cycle phase per se, we found some evidence that gray and white matter volume measures would change across the menstrual cycle in healthy females. And so what we did was we also determined the correlation of estrogen and progesterone concentration at the time of the scan with cortical thickness and white matter volume. We evaluated the menstrual cycle effects on cortical thickness in the bilateral parietal cortex, which basically we're considering previously reported menstrual cycle effects that are pretty consistent in the literature on tactile sensitivity, thermal, and pressure pain sensitivity. Some of that was our work, some of that was other work of other labs back up until 1940s, I think, was late 40s, those were discussed. And finally, we hypothesized that there will be modulated functional connectivity involving those areas with significant cycle variation in gray and white matter volume during the menstrual cycle. Now, this is critical because the way that we did this analysis was we didn't just ask, is there, you know, a functional connectivity change? We looked specifically at those areas in those individuals that we were studying, which had gray and white matter volume changes, because at the time, and still, I think, it's not quite understood why there are functional connectivity, what the relationship between functional connectivity and gray and white matter volume changes are. Um, so the methods, we had 14 healthy, normally cycling females. I believe there were 3 or 4 that were excluded because they didn't have normal, well, what we defined as normal cycles. Um, we had MRI sessions during the menstrual, follicular, ovulatory, and luteal phases using a 3 Tesla scanner. We had a 12-channel head coil at the time. They took blood draws to confirm menstrual cycle phase at each scan. And then the structural MRI scans were basically a 3D T1MP RAGE scan. Our slice thickness was 1.5 millimeters and zero point 0.9 by zero point 0.9 millimeters in plane resolution. So when you do an MRI, it goes slices through the brain, and you get these stack of slices. This is an echoplanar imaging routine, and it effectively gives you this data. That some of the data I'll discuss. The resting state functional scans was an echoplanar imaging sequence. It provided 3.6 by 3.6 millimeter resolution images with a repetition time of 2 seconds. So each brain image captured every 2 seconds. We did an interleave, 24 interleave slices of 6 millimeter thickness. There was a total of 171 volumes of functional data, and this was collected over 5 minutes and 42 seconds, approximately. I mean, if you do the math, that's what it is. During painful stimulation used for another experiment, so we had this for a different experiment, which I will present later. We delivered 20 hertz electrical stimuli to the left foot dorsum with a pulse width of 200 microseconds, which was rated 60 out of 100 on the visual analog pain scale. So this is painful stimulation. And that was done related to a previous study. So on the day of the scan, each day of the scan, we had a specific definition of menstrual cycle phase. This is important for comparison to other studies. Menstrual phase we defined to be 2 to 4 days after the onset of menses. The mid-follicular phase we defined as 6 to 8 days after the onset of menses. Ovulation was the day of or the day after a positive ovulation test, which was approximately 14 days after the onset of menses. And then luteal phase was 1 week after ovulation or about 20 days after the onset of menses. And this is obviously premenstrual. MRI data analysis, we basically used this whole brain voxel-wise effect we evaluated the whole brain voxel-wise effect of menstrual cycle phase on gray matter using what's called voxel-based morphometry. It's out of a spatial parametric mapping package, basically does spatial statistics, is the specialty of it. These are developed special packages of software that are developed specifically for MRI studies. We used, in particular, VBM8 and the longitudinal analysis part of VBM8. That allowed us to get F-stats, which are basically the results of these are significant in terms of an ANOVA. There's a significant effect. And this was, of course, because of the longitudinal analysis nature, a repeated measures ANOVA. The structural volumes were also analyzed using FreeSurfer version 5.1. And we evaluated cortical thickness using the FreeSurfer sulcal and gyral parcellations. And then we analyzed that data in R later, once we extracted the cortical thickness tables. Functional time series were bandpass filtered at 0.008 hertz to zero point 0.1 hertz. Seed-driven functional connectivity was used starting with the peak voxel of the menstrual cycle phase from effect from the VBM analysis. So that peak voxel happened to be in the left inferior parietal lobule. And then we mirrored that onto the right IPL to figure out to basically look at both sides, both networks. The left IPL is the dominant hemisphere in all of these women, as these women were right-handed. Left and right IPL are also core nodes of the dorsal attention network that you may have heard about, and I will probably make a video about at some point. Group analysis of Z-maps were analyzed using AFNI's. This is an NIH software package, 3D linear mixed effects analysis, which basically that's AFNI's software package that allows for a linear mixed effects analysis using R. So it does a voxel by voxel analysis. We confirmed that the estradiol and progesterone levels were consistent with the cycle phase in which the scan was designated. So you can see that there's maybe a slight increase in estradiol and progesterone in the follicular phase. And I only mention that because that would be the primary difference in phase effect. One other hypothesis that has existed in the literature is that menstrual phase effects, those things that you see in the menstrual phase, may be a result of falling estradiol levels from the luteal phase. So we arrive at our first figure here. This is the result of the voxel-based morphometry study. You can see here that in the inferior parietal lobule and actually also in the medial parietal part of the brain, you have changes in cortical thickness. And then what we did was we extracted the left inferior parietal lobule data from FreeSurfer, and we can do post hoc testing on that then, because you can't really do post hoc testing on this because this is just a result from it was difficult at the time, at least. You may be able to now. So what we see here is the largest cortical thickness is in the menstrual phase. That's pretty clear. When we did this analysis, we did a linear mixed effects model and specified these comparisons. It is possible that there's a difference between follicular and ovulation. It's not clear in this particular analysis. I don't think there was actually a significant difference. Some of these error bars overlap because this is a repeated measures analysis. So we had 14, so these are within effects. Kind of makes it a little bit of a weaker story to say that this happens in a population as a whole, because these are they don't really extend to the population as a whole, which most scientific studies actually don't really. That's the failure to generalize. There were also significant effects in white matter analysis, and you can really see there was also a significant phase effect in the white matter analysis, and you can really see the effects of the repeated measures here, very consistent but very small volume changes. And this is white matter in the left and parietal lobule. Free surfer makes a pretty big chunk of the white matter volume under that. And then we also found that the white matter volume in the left parietal cortex was correlated with estrogen. Uh, and these are, across the phase. So there's a bunch of there's 14 subjects here, but we had 48 measures effectively. Then which is why there are 48 dots. And this gray bar simply indicates sort of like a 95% confidence curve based on this data. Then, we looked at, again, we had hypothesized effects in the somatosensory cortex, and we did see that when pulling the bilateral somatosensory cortex together, there was a significant effect where menstrual was larger than follicular and trending to larger, greater cortical thickness than ovulation and luteal. So it seems to be that the thinnest occurs during follicular phase, but this isn't from our data obvious. It's thinner than menstrual, that's clear, but that's all we can really say. So we did an enormous number of tests to see if anything was correlated to see if not anything was correlated, to see if there was an effect of menstrual cycle phase on whole brain cortical volume, right hemisphere, left hemisphere cortical gray matter, thick volume, gray matter thickness, white matter volume, white matter left and right hemisphere white matter volume. Interestingly, there was an phase effect on right hemisphere white matter. So we did actually a family-wise error correction for this whole thing. Not Bonferroni, but probably some other family-wise error correction for the p-values. And that sort of leaves a lot of these in the dust, so to speak. Left hemisphere cortical gray matter thickness here, menstrual was greater than luteal. Menstrual may have been greater than follicular. There's the trend relationship there. And that's the whole left hemisphere of the brain. And then right hemisphere white matter volume may have been a significant phase effect, follicular being greater than luteal, ovulatory greater than luteal. And if we go back to our previous slide, we can see that ovulation seems to be the peak of white matter volume in the left parietal lobule, which may explain why there's a differential effect on the right hemisphere. That's up in the air given these data. There is a positive correlation with estradiol. Interestingly enough, brain-wise, we did not see an effect of progesterone. And we did see the effect of progesterone, or estrogen, I should say, on left parietal white matter volume. We did not see those effects. Those effects have been reported by other researchers, which we'll get into a little bit. So there's our significant effects highlighted in red. This is psychophysical data. So basically we're doing sensory testing. So the electrical detection thresholds were significantly lower. So there's more sensitivity in the follicular phase, which we've seen in other studies. And then furthermore, there was lower pain unpleasantness, which is interesting. So there's more sensitivity, but less unpleasantness during the follicular phase here, which is pre-ovulatory. follicular phase. Um, so these were the sensory data that we put out. Um, so one thing that we wanted to do was look at the seed-driven functional connectivity from the left inferior parietal lobule. So like this is the seed, the darkest color there, and then the right inferior parietal lobule. So the seed is here. What you can see here is, so these were global signal regressed. Um, or actually, no, they're not global signal regressed. They're just phase corrected, or time series corrected. Uh, there's a lot of pre-processing that goes into this. But basically, what you see here is the dorsal attention network, including the parietal lobules and some of the frontal eye areas. Um, it is not correlated with the executive control network, specifically not, and then also not correlated with the default mode network, which you can see in this slice. These are pretty much standard images. They look a little more grainy because, again, this data is from 2007. Um, so we did functional connectivity analysis to ask the question, are there any significant differences when we look at the seed-driven functional connectivity over the menstrual cycle phase and correct for all of our comparisons and everything. And what we found was that the left IPL, their seed-driven functional connectivity was greatest during ovulation compared to the luteal phase. So, and then that's specifically in this fusiform, well, it's in the visual cortex adjacent to fusiform gyrus. That may be useful or may be of interest to some hypotheses about why this was happening. Why is there greater sensitivity during ovulation? Or why is there, one, greater sensitivity during the follicular phase, which is pre-ovulatory? And then why is there greater functional connectivity during ovulation? Looking at the right inferior parietal lobule seed, we see that there is greater functional connectivity to the left inferior parietal lobule. And this might be actually superior parietal. And then also the right inferior parietal lobule during the luteal phase is greater functional connectivity to the medial prefrontal ventromedial prefrontal or medial prefrontal cortex, I should say, is greater. And that is a significant effect. So these were the only significant effects. Not surprising. We have 14 participants in the study. We would like to have gotten more. So what's some of the results? So cortical thickness, we see that inferior parietal lobule, possibly bilaterally, is greater in the menstrual phase compared to follicular ovulatory or luteal phase. We think it might be thinnest in the follicular phase. White matter volume was correlated with estradiol concentrations. There was greater electrical detection sensitivity in the follicular phase. Less pain unpleasantness during follicular phase, which was interesting to me. Functional connectivity was greater in left IPL seed-driven connectivity, and that was greatest in ovulatory phase. This is compared to luteal, but possibly also menstrual and possibly also the menstrual phase, and that was specifically in the left visual cortex. There's some supplementary data that I didn't show. Ovulatory phase functional connectivity is greater compared to follicular and possibly, again, luteal and menstrual phase from right to left IPL. The IPL is a core node in the dorsal attention network. This is also known as the task positive network, which is important for top-down, target-directed, and sustained attention. People that have lesions in this area of the brain, particularly the parietal lobe, demonstrate attentional neglect and have problems grasping things and have to go through therapy to occupational therapy to be able to work. Like, there's famous images where patients have shaved half of their face and completely neglected the other half. Um, Peak sensitivity to a broad range of stimuli. There is peak sensitivity to a broad range of stimuli during ovulatory phase. This would likely be evolutionarily advantageous as well as follicular phase, to be honest, as sperm can last in the female um, human for days. And these would likely be an evolutionary advantage for mate selection during concealed ovulation. Concealed ovulation is particular to humans. A majority, I think maybe all other species, have very obvious ovulatory signs, scents, etc. And the female human does not have those at all. Um, Visual and olfactory perception are frequently reported as most sensitive during follicular ovulatory phase, and these are important for memory formation or important adjuncts to memory, and humans are largely visual creatures. And we showed the effects of functional connectivity in the visual cortex during ovulation. The most parsimonious hypothesis of this enhanced functional connectivity between critical nodes of the dorsal attention network is that they drive an increase in a broad range of perceptual sensitivity, particularly for evolutionarily salient stimuli. In terms of humans, evolutionarily salient stimuli include faces. There's actually a face selective region of the brain called the fusiform face area. Um, and that is particularly activated in primates in general. Therefore, it's probably most parsimonious that this dorsal attention network, which really is task driven, um, is connecting with those regions of the visual cortex, particularly the fusiform face area, to co-opt and enhance sensitivity. This is a hypothesis. I think it's a pretty rational hypothesis. Um, so here, the proximate mechanism is enhanced perceptual sensitivity, and the ultimate functional result is a preference of certain facial features during ovulation that can coexist with it, and these coexist within the evolutionary framework of biology. Um, what happens, what's interesting to me is that um, we have a lot of studies that have looked at facial selectivity. Females have a tendency to, and this is a minor tendency, I will say. These are small effect sizes to select more masculine faces, faces that look more healthy. There's a lot of different preferences and biases that are enhanced during follicular and ovulatory phase. And I'm saying ovulatory phase is really like a 36-hour period. So hormone-related fluctuations in neuroplasticity would extend to influences on cognition, emotion processing, motivation, and sensitivity to disease. There have been several more findings since 2020 when we published that paper. Bilateral hippocampus and estradiol are uh, estradiol concentrations and gray matter volume in the bilateral hippocampus and bilateral hypothalamus of people that have never used oral contraception are correlated, and this is in a large sample of 37 participants. Another study has found that cortical thickness is greater in mid-follicular versus late luteal. This may include ovulation as well. Um, and late luteal follicular phase was our nadir, but it depends on uh, ultimately depends on um, the days that were chosen as definition. This is one of the issues in this literature is um, the days of what is follicular and luteal and menstrual can be overlapping. Um, and, that's something that can be taken care of in review, or at least addressed in review. Um, Another finding is larger functional connectivity from the default mode network to what looks like the parietal cortex during pre-ovulatory uh, compared to luteal. This would be somewhat consistent with our findings, though we found primarily this was ICA based, so this is a data-driven method to discover um, networks, brain networks. Um, this is pre-ovulatory is greater than luteal in the right IPL, I would assume. Ours were shifted. Our data were shifted right, left, so done in radiological format. These are, I think, in anatomical or neurological. This is a large group of 60 individuals. Um, Another finding was uh, connectivity strength was region specific in increases. So, pre-ovulatory was greater than Menzies, um, but in this case, the green blob here, which is some subcortical to cortical connectivity, possibly insular, possibly claustrum. Claustrum's very hard to image; it's very thin, and here we see connectivity strength peaking specifically in that green area, in those between those two green areas, as um, um, a function of in luteal phase. Here we have alpha band magnetoencephalography. This is related but different from electroencephalography. So we're looking at actually channel or the electrical activity of the brain. And these authors have reported an enhanced betweenness centrality in the right posterior cingulate gyrus, which is part of the default mode network, which is in opposition to the dorsotension network. Um, though these areas work together because the right posterior cingulate gyrus probably has something to do with um, autobiographical memory, for example. And then they found that the difference between periovulatory and mid-luteal between these two was significantly correlated with in this right posterior cingulate gyrus, the between the centrality was significantly correlated with the change in estradiol. So I thought that was a novel approach. It was interesting. What does it mean? They didn't explain really what they thought between the centrality meant in this paper. I thought that was a minor weakness. But again, these are hard data to analyze. In another paper that basically looked at um, um, healthy participants. It's an N of 16 in this particular task. What they're doing is they're engaged in a delayed discounting task. So they have to make a decision between taking an early reward and a late reward. And usually the late reward is larger. And so they make a decision and they have to make it relatively quickly. Um, And during the time that they're making that, you see activity in right putamen, right caudate, left putamen, largely DLPFC, but there is a cycle phase difference only when these individuals chose the sooner on the trials where they were like, I want this now. So these are impulsive, possibly impulsive decisions. There's definitely a delayed, or not, definitely there's a failure to delay gratification is what you would say, or uh, um, a preference for the more proximate reward. And the difference in activity is found just on those sooner trials, and it is specific to um, late the difference in the phase cycle effect is specific to the caudate and the putamen. Um, and that difference is they looked at late follicular phase versus mid-luteal phase. So late follicular phase is often considered to be um, can be considered to be paraovulatory. So you can see here where I've highlighted those results. I think they're different because they're in different phases. So you have mid-luteal phase here. There's a positive correlation. In the late follicular phase, where maybe there's some greater impulsivity, is the argument here, than the negative correlation. So the lower the K, the greater discounting. So there's a positive correlation between discounting and functional connectivity between the right caudate and the bilateral DLPFC during the follicular phase. Um, these This paper also had a second section of results where they looked at independent components analysis, the independent component of the executive control network, looking at the functional connectivity between the executive control network and then mid-luteal and late follicular phase. So the executive control network, there's enhanced functional connectivity between the executive control network and the right DLPFC in this case. And this is in a separate set of women. This is a cross-sectional study. And that's basically an N of 25 for the follicular phase and N of 24 for the luteal phase group. Then that functional connectivity to the right DLPFC, to the right caudate, seems to track attentional impulsivity scores. Um, so and it's particularly strong during the late follicular phase. It's not as strong of a relationship in the mid-luteal phase. Another study showed that during the periovulatory phase, functional connectivity from the dorsal ACC, which is dorsal ACC is usually right about here, to both the right orbital frontal gyrus and the right middle frontal gyrus were greater in a group of 75 individuals, 75 females, during the periovulatory phase compared to the luteal phase. So this was during listening of novel metaphors of men that it was a recording of men that were asking about whether they would want to go on a date and using these novel metaphors in particular because those are typically more attractive. And so this was enhanced functional connectivity during that task. And then finally, we have another study, and this was also looking in patients with premenstrual dysphoric disorder, which is a devastating disorder for many women. that has multiple they end up with facing multiple emotional difficulties and things like that. Premenstrual dysphoric disorder. Dysphoria is typically considered one of the least, it's the opposite of euphoria. So that gives you an idea. In the healthy individuals, it was interesting to me. This study was interesting because they show in these controls follicular phase reactivity in the amygdala to negative aversive facial stimuli. So angry people, things like that, were greater during follicular phase versus luteal phase, specifically in the left amygdala. Uh, Additional recent findings include greater functional connectivity between right posterior parietal cortex and left inferior lateral occipital cortex, which is somewhat similar, during late luteal phase compared to mid-follicular phase. So this would be also opposite of what we found. And these differences are frequently um, there are differences basically, you know, between results. Um, what does this mean overall? It's a good question. Um, there's a positive relationship between progesterone and white matter diffusion and a negative relationship with cortical thickness and progesterone in the medial and orbital frontal brain structures. This there were this positive relation there was a positive relationship between progesterone and cortical thickness and frontal eye fields and parietal regions. This would somewhat track with our study's results. And then participants in late follicular or mid-luteal phase found that greater progesterone concentrations increased accuracy in classifying female but not male faces, which was mediated by bilateral inferior frontal gyrus activity. Now that's quite interesting because that sort of is counterintuitive given any evolutionarily driven hypothesis. So the overall summary is the menstrual cycle seems to be associated in various studies with gray matter plasticity, resting brain and task-driven brain activity, resting brain connectivity, white matter plasticity, and inhibitory and excitatory brain responses. Progesterone appears to be inhibitory, and this may be mediated by, this is thought to be mediated by metabolite allopregnolone of progesterone, which binds to the GABA receptors in the brain. Estradiol may be excitatory, and it seems to enhance plasticity based on numerous human and animal studies. There's phase-specific activity changes, and plasticity may be supporting mate selection. This is a hypothesis. It needs to be tested more thoroughly, much more thoroughly. And this is likely associated with variable disease vulnerability, and macroscopic neural plasticity is possibly, I would say likely, associated with inflammation. The next steps for our lab will be a further review of the published literature, likely publish some of the previously unpublished conceptual reviews. So I need to put together the literature and see if it supports my hypothesis ultimately. And then write a grant proposal. And I think I would focus this on non-invasive brain stimulation on menstrual migraine. Menstrual's misspelled there. And related changes in brain activity in association with peripheral inflammation. We would expect that. Individual specific relationship between peripheral inflammation, brain activity, plasticity and sensory sensitivity before and after non-invasive brain stimulation would be interesting, especially in individuals that had either premenstrual dysphoric disorder or menstrual migraine, menstrual associated migraine. And here are all my references for this particular presentation. Um, and I thank you for your time.